Thank you all for coming. My name is Miguel Tam, and I'm a senior product manager with BioLegend. And today I'm going to show you some interesting data that we have collected and showing you how important it is to tackle such a complex disease as cancer from as many angles as possible to succeed in the treatment. And to start, I'm going to just show you the layout of my presentation. And I have divided the presentation into 3 different sections. And the first one is addressing the role of RANK and RANKL in cancer migration. And we look at the effect of RANKL in the cytoskeleton of the cell, and also we look at the intracellular pathways that RANK and RANKL activate to induce cancer migration. On the second section, we are also continuing working with the melanoma cell line model. And in that section, I'm going to present you some data that is looking at blocking of immune checkpoint molecules. And in this section, we use anti-PD-L1 as a treatment. And you will see some data that we have generated at BioLegend and also in collaboration with the group of Thomas Gajewski in the University of Chicago. We look at the intracellular mechanisms of anti-PD-L1 treatment as well as the cellular mechanism by which anti-PD-L1 is controlling cancer growth in this melanoma cancer cell model. And in the third section, I'm going to show you some data. It's very interesting. It's a collaboration that we are doing with Professor Siv Ronay at the Sanford Burnham Institute in San Diego. And the interesting thing with this model is that they have a knockout animal, and this knockout mouse can actually control very efficiently the cancer growth. And we look at the mechanism by which that knockout animal is doing that. And some of the things that we did was depleting specific cell types, such as CD4 and CD8 T cells. And what was the effect of that depletion and other molecules that we look at in this model in controlling cancer? Now, looking at cancer treatment, what are the strongest pillars of cancer treatment nowadays? And we often have the unavoidable classical treatment, such as surgery, radiation, and chemotherapy. But in the last 10 years or so, a new field of treatment that's been developing is immunotherapy. And by immunotherapy, we can classify actually all the treatment that is aimed at harnessing the immune system of the patient to fight that same cancer that they are suffering. And in the immunotherapy field, we have basically 3 main areas. One is the monoclonal therapeutic antibodies. The second one is the immunomodulators. And the third one is cell-based therapy. Now, in these 3 different fields, in the monoclonal antibody fields, we have a number of monoclonal antibodies that have been already used in treating cancer. For example, we have anti-CD52. That's an antibody that's been used to treat certain leukemias. We have anti-HER2. This is a very well-known antibody in the breast cancer field. We have the successful anti-PD-2. We've heard about Keytruda. This is an antibody that's used for the treatment of non-small cell cancer, lung cancer cells. And it's also been used in melanoma treatment. And we also have anti-CTLA4, which is also used in melanoma treatment. In the family of the immunomodulators, we have mostly cytokines, interleukin-2, for example, and also interferons are very widely used to treat cancers. We also have some small molecules. Some of those are aimed at activating the immune system in general, for example, the toll-like receptor agonists. These molecules are activating the immune system by engaging these receptors that are very much controlling the activation state of immune cells in the immune system. And we also have some other soluble factors that I'm going to talk about in a few slides. Now, in the cell-based therapy field, we have everything from classical transplant of bone marrow, for example, all the way to more experimental approaches such as vaccines, dendritic cell vaccines, and the CAR T cell therapy that's been developing in recent years. And throughout my presentation, I'm going to focus on monoclonal antibodies and immunomodulators, 2 fields that are very close to what BioLegend is doing. To start with the immunomodulators, I want to show you this paper that was published a few years ago, back in 2006. And the interesting thing about this paper is that it was analyzing the role of RANKL in cancer cell migration and bone metastasis. So the authors in this paper, they go beyond the classical bone homeostasis that RANKL is known for, and they look at the cancer cell migration in this model. They didn't go deeply into the molecular and cellular mechanisms by which RANKL was controlling or inducing cancer cell migration, and we wanted to look at that in our experiments. And the first few experiments that we did was to make sure that our melanoma cells were expressing RANK, which is the receptor of RANKL. And here in the first histogram graph, you see there is a histogram overlay. In blue, we have RANK conjugated to PE. In red, we have the isotype control. And then you can see that our cells actually shows a very nice separation of the positive signal versus the isotype control signal. So we can see that the cells express RANK by flow cytometry. We wanted to confirm that with another technique, and then we did a Western blot. These are two different experiments. And even when the signal might look a little bit dimmer here, you can clearly see that we see a band on the B16 melanoma cells. And therefore, we can actually confirm that the cells express RANK by two different methods, flow and Western blot. Now we wanted to confirm again the role of RANKL in inducing migration of cancer cells. And for that, we set up an in vitro assay. This is a classical bioassay experiment where we incubate the cells with RANKL on the top. This is a transwell experiment. And then we measure how many cells migrate through a protein matrix that is covering this well here. So this is a collagen protein gel, and then the cells migrate through that matrix by the action of RANKL. And then we measure the cells on the other side. And you can see here in this bar graph, when we use increasing concentration of RANKL, how the number of cells migrating through the matrix increases as the concentration of RANKL increases. Now, looking at the changes that may occur in the cell during the RANKL incubation and the migration of the cell, we look at the morphology of the cell and also the formation of filaments of beta-actin in the cell. And you can see that when we incubate the cells with RANKL, you can start to see these filaments of beta-actin moving into the cell. And actually about 10% of the cells, they completely change the shape of the cell. They look more compact. And this phenotype is probably helping the cell in migrating through the protein matrix, because they are actually reducing the area of the cell, and they can go through the pores of the matrix easily that way. Now, looking at the activation cascade induced by RANKL, which is summarized here, we can see a number of intracellular proteins that are really important for intracellular signaling. Two of those proteins are ERK and P38 in the MAP kinase's signaling pathway. And of course, we wanted to see which of these proteins were involved in this signaling pathway when RANKL was used to induce cell migration. And we focus on these two. And as you can see here in this Western blot experiment, we can see very early activation of ERK by measuring phosphorylation of the protein after 5 minutes of incubating the cells with RANKL. And then we also see increased phosphorylation of P38 at the same time point. Now, the next series of experiments that we wanted to do was, of course, to find out which molecules were which genes were being activated after incubation with RANKL at the end of the signaling cascade when NF-kappa B was activated. So the way we did that was by doing immunoprecipitation experiments, and these experiments work in a way that you pull down your sample with anti-NF-kappa B antibody, and then you analyze which genes are being bound by this transcription factor. And in this case, we did a qPCR to amplify BIRC3. And as you can see here, when the cells are incubated with RANKL, there is an increased binding of NF-kappa B to this gene. Likewise, when we use anti-NF-kappa B P65, we also detect an increased binding of this transcription factor to the same gene. Now, this is a classical gene that is activated by the NF-kappa B transcription factor. And at this point, of course, we don't have enough data to demonstrate that this is in fact a gene that is directly involved in cancer cell migration, but it is a gene that is involved in protecting the cells from apoptosis. So that's another way in which the cells might be infiltrating and disseminating throughout the body because they are protected from cell death. And we are doing more experiments to find out which other genes may be activated by RANKL through NF-kappa B activation at the end. Now, looking back at the signaling cascade induced by RANKL, we can see that, of course, this is a pathway that must be tightly regulated. There are a number of different intracellular processes that are activated through RANKL, and one of the molecules that help regulate RANKL signaling pathway is osteoprotein, or OPG, and this molecule actually can block the RANKL signaling into the cell. And of course, the next step for us was to find out if OPG treatment was able to revert some of the things that we were looking at, the intracellular signaling cascade and cellular morphology when the cells were incubated with RANKL. In this case, if we go back to the immunofluorescence experiment, we can see that when we use OPG at 10 micrograms per mil, all the rearrangement of the intracellular actin was virtually gone. We cannot see any longer the long lines of beta-actin in the cell. Similarly, when we repeated the experiments, the bioassay experiments, looking at cell migration, we can see that there is a trend in which when we incubate the cells with increasing concentrations of OPG, there is less migration of the cells. In this experiment in particular, there is a little bit of a higher background here. You can see when the cells are not incubated with RANKL, they migrate. We are still optimizing this assay, but I have no doubt that this trend is real. When you have increasing concentrations of OPG, the number of cells migrating on the other side of the well decrease. And in the same paper that I showed you a few slides ago, the authors did look at OPG affecting RANKL signaling. So when the authors use OPG as treatment during the cancer development, they can actually see that the metastasis is alleviated, there is less metastasis, and they also see that other effects of the tumor are reduced, like the paralysis in the mice. It is greatly reduced when they use OPG as a treatment. And having that said, also, there is a monoclonal antibody that is blocking the RANKL signaling. This antibody was developed by Amgen, and it's called denosumab. And the initial antibody was developed to treat classical bone function that were mediated by RANKL, such as osteoporosis, for example. But going further than that, this antibody is now being used also in the treatment of bone cancer, and the usage of monoclonal antibodies in the treatment of bone cancer, or in cancer in general, brings me then to the second part of my talk, which is focusing on the treatment of the cancer model with antibodies against immune checkpoint molecules. In this case, we use anti-PD-L1. And when we injected PD-L1, anti-PD-L1 in the cancer model, we look at the changes in the CD4 and CD8 T cells. And we also look at the cytokine and chemokine profile in these animals, in the serum of the animals. And at the end, we look at the tumor-specific T cell response and how the treatment in general was affecting the outcome of the cancer model. And Not that the immune checkpoint molecules need a lot of introduction in this audience, but just to say that immune checkpoint molecules are molecules that are hardwired in the immune system, and they are key controllers of the immune cell response. So a lot of, or several tumors can actually hijack that immune checkpoint molecule to try to escape the immune response that the immune system is going to elicit against the cancer cell. And to study that, we have a melanoma cell line cancer model. All the previous experiments that I show you were in vitro experiments. All the experiments that I'm going to show you now in this section are in vivo experiments. And we have the melanoma cancer cell line, and we implant the cancer cell lines in the animals, and then they receive a treatment of anti-PD-L1 in this case every 3 days. And after 14 and 24 days of implanting the tumor, then we collect the samples and then we analyze the phenotype of the lymphocytes in the draining lymph nodes and in the spleen. We also collect the serum of the animals, and then we quantify the cytokine and chemokine response. And we also collect the tumor itself, and then we look at the lymphocytes that are infiltrating the tumor and how the tumor microenvironment is controlling maybe the lymphocytes that are being recruited and the response in general within the tumor microenvironment. And in this first slide showing you data, you can see here that to your right is the animal that received the anti-PD-L1 injection. To your left is the animal that received the isotype control injection. And we can clearly see that when the animals receive the anti-PD-L1 injection, they have a more activated phenotype. They express higher levels of CD25, and they also express higher levels of PD-1, the receptor of the anti-PD-L1 antibody that we are injecting. In this slide, I'm showing you a summary of all the different molecules that we look to characterize the activated phenotype of the T cells. We can see that the animals that receive the anti-PD-L1 injection express higher levels of CD25, also CD69, PD-1, and ICOS. CTLA-4 remains low and basically unchanged. This is somewhat expected. CTLA-4 has a more inhibitory role in the immune response, but all the other molecules that are characteristic of an increased activated phenotype are expressed at a higher level in the animals that receive the anti-PD-L1 injection. This is looking at the CD4 T cell population in the spleen. I'm not going to show you the data, but we have similar results for the CD8 T cells in the same organ. And I also wanted to look at antigen-presenting cells. Having characterized an activated phenotype in the CD4 T cells, I wanted to see if the antigen-presenting cell phenotype was correlated somehow in their own phenotype with this activated T cell phenotype. And what I did here, you can see in the dot plots how I gated for the dendritic cells. I'm showing you here live MHC high, CD11C high classical dendritic cells. I also stained for CD80, CD86, and PD-1, and you can see here in this histogram overlay that the animals that receive the PD-L1 injection, when I look at those dendritic cells, they actually express a lower level of CD80 as compared to the animals that receive the isotype control injection. And you can see here that the animals that receive the anti-PD-L1 injection, they express lower levels of CD80. And to me, that's an interesting phenotype because that's actually supporting somehow the activated state of the T cells. CD8 and CD86, they have a different dynamics when it comes to interaction with CD28 and CTLA-4. And what can possibly be happening is that the animals are expressing lower levels of CD80 to make it easier for CD86 to interact with CD28 and then promote an activated phenotype in the T cells. And here in this slide, I'm showing you a tissue section from the draining lymph node. The previous experiments that I showed you were looking at the spleen. And here in the lymph node, you have to your left the animal that received the isotype control, and then to your right, the animal that received the anti-PD-L1 injection. In this figure, we have CD4 in green, CD8 in red, and CD11C in cyan. B220 is in blue just for contrast staining. But when you look at this figure, the combination of CD8 red and CD11C cyan give you a magenta color, a color like this one. And here in this figure, you can see that when the animals receive the anti-PD-L1 injection, there is a higher number of CD8 alpha positive dendritic cells in this draining lymph node. And this is also a very interesting result because the CD8 alpha dendritic cells, those are the ones that can cross-present to the CD8 positive T cells. And that's a major feature in cancer, in fighting the cancer in these animals, because these CD8 alpha positive T cells, they can cross-present the neoantigens in the melanoma cells and present it to the T cells, and then those CD8 T cells can kill the cancer cells. Now, having characterized the lymphocytes and the tissue response in these animals, we wanted to quantify the cytokine and chemokine response in the animals, in the serum of the animals. And we were able to do that using our multiplex system. We call it Leyenplex. And this is a multiplex assay that lets me analyze up to 13 different chemokines or cytokines in the same sample using a very small volume of sample. So that's very convenient. Using that, I could measure 13 different chemokines and many more interleukins and cytokines in the serum of the animals. These are just examples of the standard curve that you can generate using this system. And to show you the data here, this is pretty interesting because when I look at the individual cytokines, I can see that the animals that receive the anti-PD-L1 injection, here always to the left in this bar graph, they show a higher content of interferon gamma and TNF alpha, which is very specific for the Th1 response. But they also have a higher content of IL-9 and IL-22, which are associated with Th9 and Th22. So to me, the animals are actually being able to expand the T helper cells that they need to fight the cancer. And at the same time, the animals are producing less amount of important pro-inflammatory cytokines such as IL-12P70, IL-27, IL-33, and GM-CSF. And at the same time, the profile gets even more specific because the animals are able to produce certain chemokines that are probably helping in them in recruiting the cells that they need to fight the cancer, such as MAPK alpha, eotaxin, MAPK, and IP10. Now, summarizing that data, because it's easier to see that in this cartoon, I am showing you here a summary of all the cytokines and chemokines that we measured. And you can see that the animals have increased content of Th cytokines, such as interferon gamma, TNF alpha, IL-22, and IL-9. At the same time, they are able to increase the production of important chemokines, such as MAPK alpha, VLC, eotaxin, MAPK, MDC, MAPK1 beta, and IP10. But they don't have like a general response where they upregulate every single molecule that they have in the system. They actually have a very specific phenotype that includes downregulation of important pro-inflammatory cytokines such as IL-12P70, IL-27, IL-33, and GM-CSF. And finally, I'm going to show you here some data that is illustrating how the antigen-specific T-cell response behave in these animals. So all the data that I showed you before this slide corresponds to 14 days after implanting the tumor. Now, this experiment is done with animals that had been implanted with the tumor for 24 days, but also received 3 doses of anti-PD-L1. And what's different in this model is that we use a melanoma cell line that is transfected and expressing this model peptide, SIY. This is a peptide that is coming from the vesicular stomatitis virus. And it's just a model peptide that let us track the antigen-specific T cells because we can use a pentamer that is loaded with that peptide to look for the antigen-specific T cells. And as you can see here, these are CD8 T cells. And 24 days after implanting the tumor and receiving the treatment, we can see and we can track the antigen-specific T cell response. Here to your right, you can see the summary of all the animals that were in the experiment, in that particular experiment, and you can see that 24 days after implanting the tumor, there is a clear trend for the animals to expand this antigen-specific T cell response. I am not showing you here the data again, but I can tell you that these antigen-specific T cells, they also produce more interferon gamma than the cells, the CD8 T cells that are not specific for the model peptide. And finally, to confirm some of the data that's been published already using anti-PD-L1 as a treatment, we can also see that treatment with anti-PD-L1 helps the animal to keep the cancer under control. The tumor growth is a lot slower in the animals that receive the antibody as a treatment compared to the animals that receive only the isotype control injection. And having done that in that melanoma cancer model, I move on to analyzing the same cancer model, but in this case, we did some experiments with Dr. Ronay at the Burnham Institute, and the peculiar thing is that they have a very specific knockout model that is extremely efficient in controlling cancer growth. At this point, I can't reveal the identity of the molecule, but I can tell you that this knockout mouse, when they get the melanoma implanted, they fight the tumor very efficiently and the tumor doesn't develop. But then, having that model, we wanted to know which cell types were actually involved in controlling the cancer growth. And from previous experiments that had been done, we knew a few cell types that we wanted to focus on. One of them was CD4 T cells, the other one was CD8 T cells. And the aim was to deplete those cells and see what effect that had in the cancer model. And we also had, of course, other experiments designed to identify which molecules were probably involved in controlling the tumor growth in this particular cancer model. So going to the mechanisms of cell depletion, antibodies can deplete cells by a number of mechanisms. And they can do that by recruiting the complement, they can do that by cytotoxicity, they can also do that by inducing apoptosis. And perhaps the most common or the most successful antibody inducing cell depletion as a drug is rituximab. This monoclonal antibody is an antibody that is directed against a protein that is expressed by B cells, CD20. And as the molecule is expressed in B cells, this drug is extremely successful in treating diseases that involve dysfunctional B cells. That includes some lymphomas, some leukemias, and also some autoimmune disorders. And in fact, rituximab is one of the drugs that is on the WHO list of essential medicines. This is a list of drugs that the WHO considers should be in every basic health system. So it's a very efficient monoclonal antibody. Now, as I was mentioning before, in this cancer model that we have now, the peculiar thing is that when we knock down this protein in these animals, those animals are really extremely efficient in controlling the cancer growth. We wanted them to analyze which cell types were involved in controlling cancer growth in this animal model. From previous experiments, we wanted to focus on CD4 and CD8 T cells. And the first thing we did was to inject anti-CD4 antibody in these animals to be able to deplete the CD4 T cell population. And as you can see here, both in the wild type and the knockout animals, when we inject the anti-CD4, there is a strong depletion of the CD4 T cells. Interestingly, the CD8 T cell percentage goes up a little bit, maybe by compensating the system. There is also a double negative population that appears when the cancer develops. We obviously cannot deplete that cell using anti-CD4. Those are just cells that appear in the wild type when they have the tumor implanted. We don't see that many in the spleen this is spleen data, when the animals don't have the cancer. This is a cancer-specific population. But the CD4 injection is able to deplete the CD4 T cells very efficiently. And when we did that and then analyzed the surface phenotype of the CD8 T cells that remain in the system, we can see that the CD8 T cells, again, they have a strong activated phenotype. They express higher levels of CD25, CD69, and ICOS. And interestingly, CTLA-4 shows a very peculiar pattern because it is totally the opposite as what it is in the wild type. This is what you expect in the normal animal for this molecule, which has an inhibitory role, but the expression is completely different as the wild type molecule. So obviously CTLA-4 caught our attention, and we wanted them to focus on some immune checkpoint molecules again. And what we did was to inject anti-PD-1. This is not PD-L1 as the previous experiments. This is anti-PD-1. And this first slide is just showing you that there is no cell depletion when we inject anti-PD-1. There is no toxicity when injecting the antibody. You can just here see normal levels of CD4 and CD8 T cells, including our double negative cells. They are still there. But when we analyze the surface phenotype of the CD4 T cells when we inject the anti-PD-1, we again can see a very peculiar phenotype. Now, the activated phenotype is similar between the knockout and the wild type, but if we look at CTLA-4 now, we can see that the trend is reversed. Now we can see that the CTLA-4 actually behave like the wild type animal. So obviously CTLA-4 is an important molecule probably in this system, and we are doing more experiments to define the role of immune checkpoint molecules in this system. The work is now on review in the Science Journal, so hopefully it will be published soon, and we will then, there is a lot more data on the manuscript describing immune checkpoint molecules treatment in this particular cancer model. And just to summarize that final part, we can see that when we inject CD4, anti-CD4 to deplete CD4 T cells, the animals are actually not able any longer to control the cancer. They go back and reflect the wild type phenotype. That happens with the anti-CD4 injection. When we inject anti-PD-1 and anti-CTLA-4, there are also some changes. PD-1 induces some of the changes that are able to revert as well the phenotype, but there is probably more than one immune checkpoint molecule involved. So that's an ongoing project, and hopefully we'll be able to publish that soon. And to conclude my talk, I just want to say that when we look at RANK and RANKL signaling pathway, we can see that the RANKL induces actin rearrangement and melanoma cell migration. We can see that by microscopy. And we can also see by Western blot that the RANKL incubation induces activation of RANKL signaling pathway. We can see increased phosphorylation after 5 minutes when incubating the cells with RANKL. And we are now doing more experiments to modulate the pathway and see if we can control the cancer migration using OPG as a modulator. To conclude the second part, still working with our melanoma cancer model, we can see that when we treat the animals with anti-PD-L1, there is a very specific activation pattern of CD4 T cells. We can also see a very interesting pattern of cytokines and chemokines when the Th1 cytokines are being produced by the animals. They produce more Th1, Th9, and Th22 cytokines. At the same time, they downregulate some important pro-inflammatory molecules and have increased production of some of the chemokines that are helping the host recruit the cells that they need to fight the cancer. Finally, we can also see that there is an increase in the tumor-specific T cell response, and the interferon gamma produced by those cells is increased as compared to the animals that receive only the isotype injection. And we can also, of course, confirm, as it had been published before, that anti-PD-L1 treatment reduces the cancer growth in these animals. And finally, just to mention that the knockout system that we have, where the animals are able to control the cancer growth with this particular protein, we can revert the phenotype by depleting antibodies, in this case anti-CD4. This treatment, of course, is a way for us to find out the specific mechanism by which cell type and which specific molecule is involved in controlling cancer growth in this system. We are conducting more experiments using anti-PD-1 and anti-CTLA4, and hopefully that will give us a more complete answer on the whole system. And just to finalize, I want to thank some of my colleagues at BioLegend, a bunch of them that helped me put this presentation together and highlight the two collaborations we have that help us with some of the data. One is with the University of Chicago, the group of Thomas Gajewski. They help us with the antigen-specific T-cell response in the melanoma cancer model, the second section that I show you. And at the Sanford Burnham with the group of Professor Ronay, and they are the ones that developed the knockout-specific model that can control the cancer when that protein is gone. And with that, I'm going to take some questions if you have some. Thank you.